Thank you so much. This is an incredible experience. Not only because I am in Hungary again, but all these young, budding, eager scientists bodes well for the country, for science, and for humanity. I learned something today from Professor Wong, and that is that I should go back to school and to come to Hungary and to go to Szeged and come to the school and learn all these new techniques and then go work with Brian Kobilka in Stanford as a postdoc. So that's what I learned today. So when I asked me to give this talk, I really didn't know what to do. So what we're really working on is now gene editing and CRISPR-Cas and a lot of stuff on ryanodine receptor and calcium cycling. And I thought that would be a little bit rough on you guys because it's, you know. So I decided that I would talk about something if I were a student, just like you guys. And this is how I started my career as a student and working on something called adrenergic regulation of cardiac, not relaxation, of cardiac muscle. And I was in New York, and the professors there, well, they didn't know what to do with me. They said, here's the heart muscle, and see what you can do with it, right? So there were two sort of models for heart. One you took from the frog, like Ringer did, as David Eisner showed you. And the other one was from the cat. We had a lot of cats running wild in New York. So I did what I could, and that was, here is this experiment that I did in was published in 1966. You were all maybe a dream in the parents' eyes. And I isolated a little papillary muscle and measured contraction. And then I took some epinephrine and dumped it on the heart, and what really happened was contraction went up, but what I did not expect, a contraction also got speeded up. It got faster. Now, of course, that makes a lot of sense because if you're running to catch a bus, the heart is going to go faster, and you want the heart to contract faster and relax faster so you could catch a bus. Otherwise, you know, heart failure would occur. So, but you know, I was influenced a lot by English scientists. And one of the sort of big names that had won the Nobel Prize was A.V. Hill. I think he won it in 1938. And they talked about active state. They didn't understand what contraction was, and they thought that the contraction went up, you know, the active state developed very fast and stayed up there until contraction occurred. So I thought, well, you know, how do I really look at this in the heart? Because all those experiments were done in skeletal muscle. So I said, well, you know, maybe what I could do is I could depolarize the heart, right? Instead of stimulating it like this and getting single beats, if I depolarize it, that means the heart will contract and stay contracted. And that's exactly what I did here in a frog heart. So here's a frog heart that's beating, right, by itself. And then I put potassium, 100 millimole potassium, and you see that the heart goes into contracture, we used to call it, and stay there until we removed the potassium. Then I did this, what I thought was a very smart idea, namely put isopaterenol on here, and as you expect, you see, and David Eisner showed you earlier, that the contraction got twice as big. And so I was now going to show that the active state would double. Well, when I put isopaterenol on, then put potassium on, actually the heart relaxed. So I went to whoever was my mentor at that time, I don't remember, and I said, you know, what do we do with this? We're expecting this contraction to go up, just like here, but in here it relaxes. So, make a long story short, we published a paper in Circulation Research in 1966, and we called it that paradoxical effect of epinephrine. It meant that we didn't know what in the hell was going on. Later on, many years later, as a postdoc, I called this relaxant effect, and I think we were the first group to show that the major effect of catecholamines or isopaterenol or epinephrine is to relax the heart. Let that be a lesson to all of you, because you should look for that unexpected finding, right? Okay, so let's see what I did next. What I did next was this guy, Wolfgang Trautwein, which you've heard about it. He was a big professor in Heidelberg, and they had managed to use a 2 microelectrode to voltage clamp the heart. That was the first thing, and since Hodgkin and Huxley had done it in nerve, nobody had done it in the heart, and this was a big deal. And he came to New York and saw my data, and he said, you know, maybe you want to come to Heidelberg. So eventually I said, yes, I'll go to Heidelberg. And the reason I went to Heidelberg, because I wanted to know something very simple about the heart. The big question then was, you had an action potential, that is electrical activity of the heart, you put a microelectrode in, and a contraction, you see? And they're about the same duration. So the question was, the big question in the field was, did the action potential control contraction, or was it just an accidental thing, right? So I went to Heidelberg and I sort of developed this first voltage clamp technique, which was known those days as a dirty gap because it wasn't as elegant as that of Hodgkin and Huxley. And when we did the experiment in the mammalian heart, you see if I depolarize the heart and then come back very fast, in other words, just a 50 millisecond instead of 350 or 400 millisecond action potential, essentially nothing happened. The duration didn't change. But as I prolonged the action potential, now there was a regulation. So I came up with the concept that the action potential was serving both as a trigger to contraction. It triggered the release of calcium or whatever it was. We didn't know even then that there was calcium because calcium current had not been discovered. So, you know, it was 1968. And then, but after a while, it regulated it. Then we did the experiment in the frog. And what happened was, in the frog, the first 100 milliseconds didn't do anything. There was no contraction. So we looked at this, you know, there was nothing here, but then with duration, in a very linear way, control the contractions. So here was the two hearts that were the model of cardiac physiology, and they did totally different things. Totally was unexpected, right? Because frog, as Sidney Ringer had used since the time of 1880, was used as a model for cardiac physiology. So then, I was at already assistant professor or something like that at the University of Pennsylvania, and I wanted to know where the calcium was coming from. And the big question at the time was, just like in skeletal muscle, calcium was coming out of sarcoplasmic reticulum, as David Eisner told you. So the first thing we wanted to do is whether calcium was coming out of there in the frog heart, just like it was coming in the mammalian heart. And one day, I was sitting in the lab, and this guy, David Trentham, who was a biochemist, came to me. He said, you work on calcium channels and calcium channel blockers. My mother-in-law in India is having some heart problem, and her cardiologist says, get some calcium channel blocker from America. We can't get them here. I said, well, the only thing we have is nifedipine, or these dihydropyridine blockers, and they work very nicely. The problem with them is, I said, they're light sensitive. Oh, he said, maybe we can use that idea as to probe where the calcium was coming from. So here's an experiment we did. So we have an action potential and a contraction in nifedipine. That means calcium channels blocking. So the very little contraction, second beat. Then the third beat, very little contraction. Then we put a flash that lasted 400 microseconds, right, to destroy nifedipine or photolyze it, and then look to see what happened. And what happened was, the minute we did that, calcium current recovered and contraction recovered in one single beat. And you can see now the action potential is very long as compared to here, and contraction is very big. So we thought calcium was coming from the outside. Well, not so fast, because if we did the same experiment in a cat heart, something different happened. And we did this experiment here. You see the same action potential is short, contraction is very small, and then we flash. Action potential gets immediately back to normal, and it takes 8 or 7 beats before contraction recovers. And we did that under voltage clamp condition so that action potential didn't change. And you can see the same thing happen. The calcium current recovered immediately from there to there, and the contraction took 7 beats. And that's what's plotted in here. You see flash. So we said there was a beat constant. There was about 7 beats was necessary. So something in between was happening. And what was that? Well, that was because we thought calcium was being recirculated from sarcoplasmic reticulum, as David Eisner pointed out. And so we came up with this sort of scheme at the time. It's very important to have a theory to work with. And so here is the frog heart, action potential in nifedipine told you, you flash, you go from here to there in 1 beat, contraction in 1 beat. You do the same thing in a mammal. The contraction takes 6, 7 beats to get up there. Action potential doesn't change much. So we came up with a model saying in the frog, action potential causes or activates contraction by bringing calcium in, right? And then contraction relaxes by calcium maybe moving out of the membrane, calcium ATPase, or membrane sodium-calcium exchanger. Now remember, these proteins, right, were sort of recognized but not really believed, in the years that we're talking about. And in mammalian heart, well, it was calcium action potential caused calcium release by bringing calcium from the outside through the calcium channel, cause a release, and contraction happened, and then relaxation took place by calcium ATPase of sarcoplasmic reticulum, taking it back to these internal stores, or the SR stores. And we call this one the recirculation fraction. We call that the loss fraction. That's why it took 6-7 beats for this to recirculate, but it didn't happen in the frog. Okay, so now we have the two systems. Now I need to go back to my first love, right? The adrenergic regulation of the heart. So here it is, the frog, right? And here's the mammal. You've seen already this picture, and you can see this is from my doctoral thesis in '65. And when we measure contraction downward, and so when we put catecholamine on, you see the contraction really gets very, very big. Action potential hardly changes in the mammal, and relaxation gets very fast. In the frog, doing it with Jim Weiss, who was a medical student in my lab, who became the chief of cardiology at UCLA, I don't know, for 20 years. Anyway, when we put epinephrine on a piece of the frog heart, action potential got very big and got higher, and contraction got very big, right? So then is this slide, which you've already shown. I showed you paradoxical effect, remember? Okay, so here is the frog heart, and this is from a paper in '69 in Science. I think that was the only single author paper I ever had in Science in my life. Anyway, so then here is contraction in the frog heart. We depolarize, and it got very nice, beautiful contracture, and then we wash it out, came right back. Then we put isoproterenol on, and isoproterenol enhanced the contraction, doubled or tripled it. And then when we did with KCl, depolarization, well, it relaxed. We did the same thing in a mammal. Same thing happened. I wasn't expecting that, but it happened. So we now, and you should also, knowing what I've told you already, in this case, we have these sarcoplasmic reticulum, calcium ATPase, SR uptake, and all that stuff. So you could say, well, isoproterenol enhanced calcium uptake system, which it is done by phosphorylation of phospholamban and enhancement of SERCA. And so that makes a lot of sense there. But in this case, well, that was difficult to explain. And, before I did that, we did the same thing with voltage clamp. It had nothing to do with KCl. It's doing the same thing whether you have KCl or just voltage clamp. You see that action potential produces a small contraction. You put isoproterenol, you have a big, big contraction. But if you kept the depolarization on just like you kept it on with the KCl, you see that actually epinephrine relaxes instead of, so my thought on this one was that calcium current is enhanced by isoproterenol. That's how it's a paper in J. Physiol in '81. I think I was maybe 2 years old. No, just kidding. So anyway, the point was that the KCl wasn't the thing that was causing this relaxation factor. It was something else. So what was that something else? Okay. Aha. So back to structure. So by this time, in early 70s and or late 60s, people did ultrastructure of both mammalian and the frog heart. And something interesting showed up. In the mammalian heart, as David told you earlier, there is these T tubules, there's sarcoplasmic reticulum, there's the SR and all that stuff, all the relaxing machinery. In the frog heart, well, there was very, very primitive sarcoplasmic reticulum, and people that did the molecular biology found no calcium ATPase, no circa, no nothing. So in this scheme that I showed you earlier, if you put iso in here, you got calcium current got big, contraction gets big, and then it relaxes. But in the absence of any circa or phospholamban, we didn't know how the relaxation in the frog heart was happening. So, and you can see in here, right, with circa enhanced phosphorylation of phospholamban, this recirculation factor increases and relaxation occurs faster. So now I got invited to go to Buenos Aires in Argentina to give a talk. And I'm giving this exact talk that, oh, so sorry, this is just sort of summarizing this thing for you. So we have this thing that in the mammalian heart, adrenaline phosphorylates the calcium channel, the ryanine receptor, and so calcium influx goes up. That's a positive inotropic effect. It means that contraction gets bigger. And in adrenaline also phosphorylates circa phospholamban complex that stimulates calcium uptake into the SR, and that's the adrenergic relaxing factor. So in a mammal, we were pretty sure we had the story right. But what about in the frog? Well, in the frog, we had positive inotropic effect was happening the same way we talked about. Calcium current gets bigger by phosphorylation of calcium channel, and so we have this contraction goes up, and once calcium current is inactivated, the relaxing effect takes over, predominates. So you could say that in a steady state, right, adrenaline really relaxes the heart and after it potentiates it. So in the absence of significant expression of SR and circa phospholamban in the frog heart, how in the hell is the relaxation similarly enhanced? So I'm giving this lecture. I didn't ask this question, but one of the students, just like you guys, asked this question. He said, Dr. Morad, you just told us that circa and all that, but there's no circa and all that stuff in the frog, so how is the relaxation enhanced? So I went back to Penn and my postdocs. So there was this wonderful Chinese postdoc in my lab called Jin Fang, and there she is, right there. And Yaroslav Shuba from Ukraine, who's now a professor of physiology in Kyiv. So I said, well, you know, guys, we have this suppression of frog heart enhancement, and this appeared in PNAS in 96. Let's find out whether, in fact, isopaterenol is regulating the sodium-calcium exchanger, the thing that takes the calcium out of the heart. Oh, everybody argued that there are, Dr. Morad, there's at least 20 papers in a mammalian heart that shows that sodium-calcium exchange is not phosphorylated, is not regulated. So how is this happening? So we got a single heart cell from frog, and then we tried to measure the exchanger current. And I won't bore you with the technique here, but anyway, so we depolarized first, activated calcium entry, and then we would voltage clamp or ramp clamp to see what the current voltage relationship of the sodium-calcium exchanger was. And all you need really to know is that when we measured the sodium-calcium exchanger, and we would put 5 millimole nickel, and that would immediately block it. And you can see when we put ISO on, ISO suppresses it. Totally unfound, a new novel finding, and couldn't figure it out. And there's the current voltage relationship in control and ISO-treated preparation. And I immediately realized that the sodium-calcium exchanger must be somehow either different or is being regulated differently. So I knew Professor Carafoli, who was in Zurich those days, and that's Professor Carafoli, you see. And I think this is a picture from a Gordon conference that I sort of chaired, and he was there, and then there was Yaroslav Shuba from Ukraine, and Dr. Oz from Dr. Anyway, we did this thing. We cloned. He helped us clone the sodium-calcium exchanger, and it turned out that sodium-calcium exchanger in the frog heart had this 9 amino acid Walker A sequence which bound cyclic AMP, and it was known as to be as a secondary sort of way of phosphorylating the molecule. And so that is how it was being done. And then we took this P-loop 9 amino acid and inserted it in the dog exchanger and looked to see what was happening. So then you can see the dog exchanger now has this P-loop, and to our not surprise, but more or less amusement, we found that if you took the wild dog exchanger, you put nickel, it blocked it, and you put cyclic AMP or some phosphorylating agent, and nothing really happened. And if you took wild frog exchanger and you put the cyclic AMP on, 8-bromocyclic AMP, it suppressed it beautifully. And if we take the dog and we made a doggy frog chimera so that we had everything was dog except that Walker A sequence, and you can see now again we have beautiful suppression just like that. And we looked also at the phosphorylation site that was in there also, and if we knocked that out also, it stopped doing it. So by going to Argentina, giving this lecture, the student asking me this question, it resulted in this paper that we published, and then of course we did the structure and all that stuff, and found that the frog exchanger motif was this Walker A 619 amino acid, and if we put it in the dog, and the dog also had this relaxant factor. Well, I lived in Philadelphia, and when I left UCLA and went to Philadelphia, I spent one summer there. It was hot and humid, I said, I can't live like that in the summer, right? So I found out that there was a marine biostation, right? You have to be creative in life, yet you have to also live, right? So I wanted to spend my summers at a nice, cool place, and the marine biostation in Maine allowed me to go there, and so we worked on the shark heart there. And there's, this guy is me, oh, sorry, and oh, God damn it. Yes, okay, there. So, you know, we'd grab the shark, and we would take the heart out, and the shark had one ventricle, very beautiful ventricle, and two atrias. So we thought nothing else to do but to just repeat the thing in the frog heart, and that we would get the same result. Well, again, there was a curveball. There was, it turned out that the results weren't quite the same, that you had actually, again, it was published in PNAS in 2001. This postdoc of mine, now professor of physiology and a chair and a dean in Korea, National University. Anyway, you see that, again, the suppression of influx of calcium was enhanced. I mean, it was suppressed, but the efflux was either not changed or enhanced. So we start thinking that maybe there was a bimodal regulation. That is, the influx of calcium and the efflux of calcium were separately regulated. So then we wanted to know what was the molecular determinant of that. And then we found out, we cloned the exchanger, and we found out that there was a 51 amino acid insert in there where it was close to the calcium binding domain, and that if we remove that, it would look like a frog, but if that was intact, you see the situation of enhanced influx, enhanced efflux, and suppressed influx. Efflux was enhanced, influx was suppressed. And you know, we did that same thing and looked at phylogenetic aspects of it. So it turned out that different animals having expressed sodium-calcium exchanger, where they don't have sarcoplasmic reticulum or all the phosphorylation aspect of our heart, have different way of regulating calcium channels, of sodium-calcium exchanger. So I want to sort of summarize for you guys, just in case you get interested in looking at sodium-calcium exchanger. In a mammalian heart, potentiation of contraction and enhancer are mediated by phosphorylation of ryanodine receptor, phospholamban, and calcium channel. Development of SR, circophospholamban, in the mammalian heart removes the need for adrenergic regulation of NCX. So you don't want NCX, which dump the calcium out, to fight the sarcoplasmic reticulum. You know, Lady Nature knew better how to do this. So in the frog and lower vertebrate hearts, and even in embryonic mammalian heart, and I didn't show you that thing, so if you look at neonatal embryonic hearts, where intracellular calcium stores are minimally expressed, adrenergic enhancement and contraction is mediated by phosphorylation of calcium channel, But enhanced relaxation appears to be mediated by NCX. So that's very critical. How well this hypothesis holds out in different species and development remains to be tested by you. So I need to thank a few people for this work. And these were my students. He was Jimbo at 22 at Penn, medical student, became the chief of cardiology at UCLA for 20 years. And Yale Goldman is a member of National Academy, director of the Penn Muscle Institute. We used to call him Goldie because he had this sort of golden hair. And Lars Kleiman from Denmark was a professor of cell biology at University of South Carolina. And then finally, I want to pay a homage to our mentor, Sir Andrew Huxley, was very important in my life. Not so much telling me what experiments to do, but encouraging me in looking at the evolutionary aspect of EC coupling. And here he is in his 70th birthday in my favorite place in the world, in Maine, with Yale Goldman, my student at the time, whom I sent actually to his lab at the University College in London to be trained there. So I thank you very much for listening to this funny story.
Thank you very much for your presentation. We just have time for only 2 questions at most. So does anyone have any pressing questions that they would like to ask Professor Morad? There's
Yes.
one question over there.
There's a
Let's wait for the microphone.
young scientist here that wants to have a question. Now, please speak in the, yes, otherwise I can't hear you. Yes.
I might have misunderstood something, but by suppression of the NCX, yes, channel, how is the calcium removed from the cardiomyocyte? So how does the relaxation occur if the channel that removes calcium from in the intercellular space is blocked, is suppressed?
A very good question, right? There's another thing. I have to go back to the lab to do the experiment on that. But, so the calcium is coming in, right? And the calcium is going out on the NCX. But when you have isoproterenol there, there's a lot more calcium is coming in, so the influx of the calcium is suppressed. So that much calcium isn't coming in, and so the regular NCX isn't completely shut off, it's just suppressed, regulated. Right? So not enough calcium is coming in, and so that's why the contraction, is relaxes. But it's a good question.
Very nice. So, any other questions? There's one more. Well, we have just time for the last. This is the last question. I'm sorry about this.
We have to be fair to the males.
Yes,
in this,
we have to keep a balance.
Yes.
So my question is, how did you source the cat hearts for all these experiments?
How did I what?
How did you source the cat hearts? Where did you get them from?
Where did you get the hearts from?
The cat hearts.
Oh, there were a lot of wild cats those days in New York. This was, remember, it was in the 60s.
You caught them from the
No, I
street?
didn't catch them. There was an animal facility. It was, actually, I had difficulty working on it because when I went to Heidelberg, I didn't want to work on the cats, and the Germans were very hands off of the cats and the dogs. So we ended up working on sheep part, which we'd go to the slaughterhouse, get the sheep heart and bring it to the lab and make the preparation.
Thank you.
So, it's absolutely right.
Okay, so with this, unfortunately, we have to close the discussion, and we'd like to hand over the certificate of appreciation to Professor Morad, and thank you very much for coming to
Absolutely.
Szeged.
Yes.